Hello everyone, I hope you're all doing well. My name is Claudia Ramos and I'll be talking about my ongoing project, Gene Ontology Analysis of Long-Term Alcohol-Induced Neuroadaptation. The PI of this project is Dr. Alfredo Getzi and our fellow student researcher is Corinne Crosland. The hypothesis we developed is that using gene ontology as a tool, we can discover a gene network that activates when flies are exposed to ethanol vapor. So what's so important about this project? We want to discover new gene categories related to these activated pathways. We hope this study sheds some light on how alcohol affects the brain and how to create possible new therapeutics for alcoholism. As an introduction, I'll start with some common terms and definitions so you can understand the next slides with more clarity. For those of you that aren't familiar with the term gene ontology, it's a structured, controlled vocabulary for a classification of gene function at the molecular and cellular level. The main tool used in the analysis was DAVID, a web-based functional annotation tool. DAVID works by categorizing genes with annotation terms, in other words, by their biological function. And last but not least, what is an annotation term? It's a word or phrase that determines a certain biological function for a similar set of genes. Now, focusing on what's written on the slide, alcohol use disorder, or AUD, is one of the top 5 risk factors for disease, disability, and death. Alcohol is a neurodepressant, so it can disrupt the normal activity of the brain, causing it to try and recover its homeostatic function. Now, alcohol dependence or addiction stems from long-term adaptations in neural pathways that are related to habit formation, reward, dependence, and tolerance, or in other words, ethanol-induced ethanol tolerance. What's still unclear is how alcohol disrupts the homeostatic function to the point of perpetuating an addicted state. The actual experiment consisted of 400 flies total, 200 control and 200 experimental. The equipment used in the lab was very similar to the image displayed on the right, where some modified test tubes contained the flies. 5 minutes of water vapor were administered, then 30 minutes of ethanol vapor, and afterwards 5 minutes of water vapor again. 2 hours after the exposure to ethanol, the flies were frozen in liquid nitrogen and dry ice. After this, every 24 hours for 7 days, certain quantities of flies were dissected and the RNA was extracted. Because of an organization oversight, the day 5 data wasn't collected, so only 6 time points were analyzed. These random RNA sequences were stored in FASTQ files, meaning they were very compressed but ready to go through a bioinformatic pipeline. This pipeline consisted of 7 steps total, of which I'll go through each of them briefly. FastQC serves as a quality assessment, after which Thermomatic was used to trim low quality reads. FastQC was used again to see the overall quality after the trimming. Then HiSat2 was used to map the sequences to the fly genome. After this, we used SAM tools to reduce the size of the files so they could be stored locally. Then StringTie assembled the RNA sequences into possible transcripts. And finally, the ball-gown protocol was used to produce a ranked list of differentially expressed genes, which was the input for DAVID. Some additional parameters had to be put in place because DAVID's total output was 4 functional annotational results, because 2 tools were used for both the upregulated and downregulated genes. This was an exorbitant amount of information for one presentation, so we had to narrow it down. Firstly, only the upregulated genes were fully analyzed. Around 5 databases were used to classify all the genes, so we filtered it down to one database, called GeoTerm BP Direct, where the BP stands for biological process. From this single database, the remaining GeoTerms were ordered by fold enrichment, and the top 10 were chosen with some exceptions. Before I get started with the results, I'll only be commenting and highlighting on GeoTerms that have previously been identified as ethanol related, plus some other new categories. These were the results for day 1. The top 2 terms were highlighted because of their importance in alcohol response. Most genes involved in lysosomal transport can also be involved in synaptic vesicle transport, which means that they have the potential to modulate synaptic transmission. So we can infer that the ethanol is causing different synapses to activate in the brain. Moving on to the second term, it's known that alcohol responses are dimorphic, meaning they affect females and males differently. It was shown recently in Drosophila that sex determination genes are critical for alcohol-induced aggression. For day 2, we see sex differentiation is still being overexpressed with nearly the same fold enrichment of 22. Some other overrepresented genes are essential to the regulation of notch signaling pathway. This is a highly conserved pathway that its alcohol-induced activation and the accompanying transcriptional responses in memory circuitry contribute to addiction. In addition, another term, circadian rhythm. Many circadian clocks have been implicated and have a strong relationship with ethanol responses, and alcohol is also known to affect sleep, a process that is under circadian control. Moving on to day 3, our first enriched term with a fold enrichment of 34 is synaptic target inhibition. The two genes we see here are involved in the determination of synaptic target specificity, more exactly during new synapse formation, so we can infer that the exposure to ethanol is inhibiting new synapses to form. Now, if we take a closer look at the chart, we can see 3 terms: humoral immune response, response to bacterium, and defense response. These are related to the immune pathway not only in flies but in mammals too. Many recent studies have established a strong link between alcohol exposure and the immune response. It's already been shown that alcohol can alter the activity of the innate immune system, and these changes in activity can have an influence in alcohol-related behaviors. We decided to include these terms past the top 10 in order for you to get a clear and more accurate picture of what was happening on this third day. From the last slide, we saw that ethanol was inhibiting some synaptic target, but in this one, synapses are undergoing assembly, which is curious because it means that it's influencing some synapses negatively and others positively. Here, we can see even more responses by the immune pathway. In day 4, we can still see some immune system activity looking at the 3 genes belonging to defense response to fungus. Apart from this term, we see neurogenesis in the last row, and genes involved with this process, like in day 3, help create new synapses. Comparing day 3 and day 4, lots of similarities can be seen, which is interesting after seeing such an abrupt change in terms from day 2 to day 3. Here in day 6, sex differentiation makes another appearance with a consistent fold enrichment of 22. Some new interesting terms have to do with light processing and phototransduction. At this last time point, an immune response is still visible, with a fold enrichment of 22, whilst in day 3 it was 24. The gene ontology term is the same, but the genes are varied. Some are repeated, like TOTA and YP3, but the rest are different. Proof of how multifunctional genes are can be found in the TOTA gene, which is also present in the cellular response to UV. Every day is variable and different. The geo terms are diverse, but some are repeated and consistent various times, like sex differentiation and the immune response. Various categories validate previous studies, but some new ones have risen that weren't identified before, that may give us insight in long-term alcohol-induced neuroadaptations. These new categories open up investigations in other fields like psychology and other areas in neurobiology. Additional studies can now explore these new categories and the genes within them, including the downregulated genes previously mentioned. Genetic mutation to these genes can be done in the future to possibly reverse to some level the compensatory adaptations the brain underwent in its addicted state. A long-standing goal of researchers who study long-term alcohol-induced neuroadaptations has been to develop new therapeutics to treat not only alcoholism but other human conditions and brain disorders. We hope this study is one of many that continues to contribute to this goal. Thank you for all your attention.